Buddy, we're going to kick off in just a minute once everyone comes into the room. Great to have you all here. All right, I think I'm going to go ahead and get us started. So, hello and welcome everyone. Thank you for joining Southern Model Organism Corp, SMOC for short, for our second webinar of 2026. I'm Dr. Alex Sercel, a specialist in mitochondrial bioenergetics and director of West Coast Business Development here at SMOC. And I'll be today's host and panel discussion moderator. I'm joined by our panelists, SMOC's Chief Business Officer, Dr. James Jin, our Senior Vice President of Antibody Discovery, Dr. Lei Chen, and our Chief Executive Officer, Dr. Dongzhao Feng. If this is your first time meeting us, SMOC has 26 years of experience in generating genetically modified rodent models, providing custom gene editing services, and over 18,000 ready-to-use animal models, including more than 1,000 humanized therapeutic target strains, which will be the subject of today's webinar. Built on our expertise in and catalog of genetically humanized animal models, we also provide a comprehensive suite of full-service CRO offerings across autoimmune, oncology, metabolic, and neuropharmacology indications, in addition to SMOCmab, our fully humanized antibody discovery platform, which we discussed in last month's presentation. Please reach out to anyone from our team for a link to that talk. Without further ado, it's my great pleasure to introduce today's speaker, Dr. Shuang Li. Dr. Li is the study director of R&D at Shanghai Model Organisms Center in Shanghai. She earned her MS from Peking University and her PhD in medical immunology from University Libre of Brussels, where her research focused on the critical role of non-inherited maternal antigen-specific T cells in maternal fetal immunity. After returning to China, she worked as a research scientist in antibody drug discovery at Hengrui Pharmaceuticals and ReamGen, specializing in oncology and autoimmunity and gaining deep hands-on experience in preclinical pharmacology and efficacy assessment. At SMOC, Dr. Li leads the development, validation, and application of a wide range of humanized mouse models for preclinical pharmacology, efficacy, and safety evaluation. Please type any questions for our speaker and panelists into the chat, and we'll address them in a live Q&A session after the presentation. So please stick around until the end of the webinar. Dr. Li, the floor is yours.
Hi, everyone. Thank you for joining today's webinar. My name is Shuang, and I am a study director at SMOC. It is a great pleasure to be here with you today. And the title of my talk is SMOC Humanized Mouse Models, Bridging the Gap Between Discovery and Clinical Success. We will start with the general introduction of humanized mouse models. Then we will dive into 4 major application areas. Part 1 covers oncology, Part 2 focuses on autoimmune diseases, and Part 3 explores small nucleic acid drugs, and Part 4 covers PK studies. And so in each section, I will present several representative models as examples. On this slide, you can see the global biologic market is growing rapidly with a projected compound annual growth rate at 10.76% from 2026 to 2034. It is expected to expand from around $459 billion US dollars to over $1 trillion US dollars. This market covers antibodies, vaccines, recombinant proteins, antisense, and RNA therapeutics. These drugs are engineered for high specificity and potency against human targets. So this brings us to focus on the amino acid sequence conservation between human and mice. And human mice sequences are not identical. Certain residues differ, and variations within or near an antigen epitope can dramatically impact antibody binding. Subsequently, a therapeutic antibody may fail to effectively recognize or engage the mouse target. Well, non-human primates are genetically much closer to humans, which is why many companies adopt them for preclinical studies. However, They come with high cost and limited supply. And human mouse models provide a practical alternative. And here we summarize different preclinical evaluation approaches. In vitro assays are low cost and fast, yet they lack a functional in vivo immune system and deliver limited translational value. Conventional mice are rats, and rats are affordable, offer reasonable throughput, and are easy for the genetic engineering. Still, their relevance to human targets remain limited. Non-human primates score high on translational value and have an in vivo immune system, but the cost is extremely high, throughput is low, and genetic engineering is difficult. While humanized mice carry a human target, retain a functional immune system in vivo, and allow high throughput studies and support genetic engineering. Cost is higher than regular mice, but far below non-human primates. Translational value is high, and so humanized mice offer a strong balance across these factors for preclinical assessment. This slide shows the core value of humanized mice in drug development. Wild-type mice carry mouse gene and protein, and their immune system is also mouse-divided. That is why drugs often fail to engage properly here, and leading to more clinical outcomes and weak prediction of human response. While humanized mice carry human gene and protein, or a human immune component. And this creates a better match for human drugs. And the bridge image below is intentional. It represents the value of death between drug discovery and the clinical success. The humanized mice help cross that gap. They are readily available, more cost-efficient than non-human primates, have immune system, and support high throughput and deliver high translational value. And the goal is improved success and better outcomes in the clinic. A humanized mouse refers to mouse that carries some aspects of human biology. For instance, humanization could refer to a mouse expressing a human gene introduced as a random transgene or targeted knock-in. And it can refer to a mouse expressing human immune cells, tissues, tumors, a microbiota, etc. In human gene humanized mice and the human immune system humanized mice are two main types used in clinical evaluation. At SMOC, we have developed both types. While immune system humanized mice have some limitations. For example, not fully immunocompetent, donor-to-donor variability, and the graft versus host disease. Well, gene-humanized mice keeping the mouse fully immunocompetent, and these are widely used across many diseases. And so today we will focus on gene-humanized mice and talk you through how they are applied. So we start with oncology as it stood out as the absolute global biologics market leader among major therapeutic areas. This is a landscape view of the major biologic therapeutic modalities in today's oncology pipeline. First, T-cell engager, typically bispecific antibodies that simultaneously bind a tumor-associated antigen on and CD3 on T cells, and bringing the two cells together to drive tumor cells killing. And second is the immune checkpoint inhibitor or co-stimulatory antibodies. These targets pathways that regulate immune cell activity, either blocking inhibitory signals that suppress anti-tumor immunity or activating stimulatory signals to enhance T cell responses. And third is the therapeutics that target or remodel the tumor microenvironment. This includes approaches aimed at reversing immune exclusion and modulating stromal or vascular compartments. And fourth is the agents that directed at tumor-specific or associated antigens. These modalities are biologics, and many demand high specific target recognition that creates the need for humanized mice. And we have models for most of these targets. And today I will highlight two commonly used ones, starting with T-cell engagers. According to article published in Nature Reviews, there are over 200 different bispecific molecules involved in more than 300 clinical trials. It's exciting to note that 50 of these bispecific antibodies are either in phase 2 or 3 of development or have already been approved. And when it comes to the focus of these developments, approximately 73% bispecifics are being developed to treat solid tumors. And this area is largely dominated by T-cell engagers, which make up about 33 of the bispecific antibodies. And on the other hand, for blood cancer, T-cell engagers are even more prevalent, making up about 75%. For market perspective, the bispecific antibody market was valued at around $7.8 billion in 2022. And looking forward, this market is expected to grow at a compound annual growth rate of approximately 11.2% during the forecast period from 2023 to 2030. And this growth is quite significant and highlights the expanding role and potential of bispecific T-cell engager in cancer treatment and beyond. And now let's look at how a CD3 bispecific T-cell engager works. It simultaneously binds to tumor-associated antigens on tumor cells and CD3 on T cells, cross-linking these two cell types to form immunological synapse. And this synapse promotes T-cell activation, leading to the secretion of inflammatory cytokines and cytolytic molecules that kill the tumor cells. Additionally, CD3 bispecific antibodies can recruit immune cells into the solid tumor microenvironments for cancer immunotherapy. And this is the therapeutic approach. In the therapeutic approach, the CD3 complex is one of the key molecules to modulate T-cell function. And the structure of the CD3 protein includes an N-terminal extracellular domain, a transmembrane domain, and a cytoplasmic tail region rich in immunoreceptor tryptosine-based activation motifs. It is composed of 4 different subunits, and these subunits interact to form the CD3 complex, which associated with the alpha and beta chain of the T-cell receptor, and together forming a complete TCR-CD3 complex. And this complex is important for T-cell development and functional signaling transduction. Why we need to humanize CD3 EDG? As we mentioned above, laboratory mice are excellent experimental animals. However, due to the poor cross-reactivity with mouse CD3, preclinical research into T-cell engager is hampered by the lack of appropriate in vivo mouse models. And one approach is to use immune system humanized mice. And these mice have a donor-derived human immune system that includes effector T cells. However, several types of immune cells cannot develop and mature normally in these mice. And so this model cannot be used to assess combination therapies that target the innate immune system. And they have another limitation, such as donor-to-donor variability, and the graft versus host disease. And another approach is to establish transgenic models expressing the human CD3 complex. Here, we developed a fully humanized CD3 EDG mouse models for evaluating T-cell engagers. And here is the human CD3 EDG knock-in mouse model generated in B6 background through stepwise targeting via ES cell-based back transgene. The mouse CD3 EDG gene tandem, which is located at the same allele chromosome 9, was completely replaced by human CD3E, CD3D, CD3G gene tandem. And this slide shows how CD3 EDG humanized mice can be used to evaluate both efficacy and safety of CD3 bispecific T-cell engagers. T-cell activating therapies can be associated with the immune-related adverse events, most prominently in systemic cytokine release that may progress to CRS. Here, the humanized mice were engrafted with MC38 tumors, which overexpress human EpCAM, and treated with OKT3 or an anti-CD3 EpCAM bispecific antibody. And the bispecific antibody achieve about 83% TGI with no significant cytokine release. While in contrast, the OKT3 promoted tumor growth with TGI around minus 80%, likely reflecting activation-induced cell death of T cells and induced CRS-like cytokine elevations. For example, here, IL-2 and TNF-alpha peaking around after the first dose, and interferon gamma, IL-10, and IL-5 are actively required and safety. So preclinical models need to capture not only anti-tumor efficacy, but also early cytokine dynamics. And these data support our humanized CD3 mouse models as a practical model for evaluating T-cell engager efficacy and also safety. And this slide moved to B-cell depletion using CD3 bispecific T-cell engagers. Here, glofitamab and surovitamib in CD3, CD19, CD20 triple humanized mice. And CD19 and CD20 are well established for B-cell malignancies and increasingly for autoimmune disease, where B-cell depletion is therapeutically beneficial. And to evaluate this in a more human-relevant setting, we used humanized CD3 mice carrying human CD19 and CD20. According to the results, we retains B cells in bone marrow and spleen, while both bispecifics markedly reduce B cell population. And also in peripheral blood, the vehicle retains B cells, whereas both bispecifics rapidly reduce B cells with near depletion by day 3 to 5. And now let's move from T-cell engagers to the next big area in immuno-oncology, immune checkpoint pathways. This figure depicts immune regulatory molecules in the tumor microenvironment among tumor cells, T cells, antigen-presenting cells, and macrophages. It lists co-stimulatory targets such as CD28, OX40, 41BB, alongside co-stimulatory targets, co-inhibitory targets, including PD-1, CTLA-4, and co-stimulatory region promotes T cell activation, while inhibitory pathways suppress anti-tumor responses and mediate resistance. Current immune therapies target these axes via checkpoint blockade or agonist engagement to enhance T cell mediated tumor killing. And among these targets, approved therapeutics exist only for PD-1, PD-L1, CTLA-4, and LAG-3. The PD-1 inhibitor market illustrates the scale of this opportunity, projected to rise from roughly $49 billion in 2025 to over $200 billion by 2035. And the commercial backdrop driving continued investment in PD-1 and next generation checkpoint strategies. And next, let's look at PD-1 inhibitors. And on the left, there are examples of approved PD-1 monoclonal antibodies, such as Pembo, which is also called Keytruda. And their mechanisms is blockade of PD-1 on T cells, preventing engagement with PD-L1, PD-L2 on tumor or immune cells, and thereby disinhibiting T cell activity. And clinically, they span many indications such as melanoma, non-small cell lung cancer, and so on. And the key preclinical challenge is that many FDA-approved anti-PD-1 monoclonal antibodies do not cross-react between human and mouse PD-1. And so standard murine PD-1 cannot be used to evaluate human antibody pharmacological in vivo. And that's why we developed the PD-1 humanized mouse model, in which we replaced the entire mouse PD-CD1 gene with a human counterpart. And we validated the human PD-1 expression not only on the genomic level, but also on the protein level via flow cytometry. And moreover, we tested the commercially available PD-1 antibody drug on these mice and got efficacy data as expected. The PD-1 humanized mouse is one of the first few humanized models we developed back to 7 or 8 years ago, and this model was used in the preclinical study for one of the first few PD-1 antibody drug that received the China FDA approval. And this study was published on the journal Antibody Therapeutics back to 2018. And now we move to part 2, autoimmune diseases. And this is another major therapeutic area with high unmet need. And it is the second largest segment of the global biologics market after oncology. Autoimmune diseases arise when the immune system launches an inappropriate response against self-antigen and resulting in tissue damage. These are about 100 such disorders, split into systemic diseases like lupus and organ-specific diseases like psoriasis. With over 500 million patients global, the autoimmunity ranks as the third largest chronic diseases category after cancer and the cardiovascular disease. The AD, psoriasis are among the most common, each affecting 2 to 5% of the population, depending on the condition. And over the past 25 years, modulating cytokine signaling to target the immune system has been the cornerstones of therapeutic innovation in autoimmune disease. TNF inhibitors such as infliximab have become standard of care across multiple indications. And looking at the pipeline, you can see the broadening footprint across this disease with IL-17, IL-23, JAK, and TNF family targets prominently presented. And by 2030, targets biologics are expected to represent 82.1% of the autoimmune disease market. This is precisely why humanized models are essential to faithfully engage these human cytokine pathways and targets in vivo. We also have lots of success in constructing humanized models for autoimmune disease research and the corresponding therapy development. For instance, here we use IMQ, which is a ligand for toll-like receptor, to induce psoriasis on IL-17A and F double humanized mouse model. And we measured a couple of clinical and histological parameters without treatment of test articles or positive drugs. And the data shows that this double humanized model developed IMQ-induced psoriasis like pathology and response to the positive drug, with clear improvements in clinical, histological, and skin thickness readouts. So the model can be used for evaluating human IL-17 target therapies. And part three is more nucleic acid drugs. This is one of the most rapid expanding therapeutic classes in recent years. And many of these therapeutic acts on human-specific targets, which means human mouse models are essential for preclinical evaluation. The small nucleic acid drugs have advanced very rapidly in recent years. They have moved from early proof of concept studies into a period of clinical validation and commercial growth. The field began in 1978 with the initial concept, reached a major milestone in 2018 with the approval of the first siRNA drug, and has since accelerated sharply, especially after the GalNAc delivery technology matured and enabling efficient targeting to the liver. The pipelines are expanded dramatically. Indications now go far beyond rare genetic diseases, reaching obesity, NASH, Alzheimer's disease, and increasingly oncology and disease target non-liver tissues. And this is no longer an emerging concept. It is a therapeutic platform entering full industrialization. And the main type of small nucleic acid drugs include ASO, siRNA, mRNA, etc. And if we look at the pipeline share, the ASO and siRNA together make up about 70%, or roughly 38% for ASO and 32% siRNA. And these two are the most clinically advanced and also the ones that we see most often in our efficacy studies. ASO is a single strand of DNA or RNA, and it binds into the complementary mRNA sequence. This recruits an enzyme called RNase H1, which cuts the RNA strand. As a result, the mRNA is degraded and the protein translation is blocked. While the siRNA is a double-strand RNA, it recruits a complex called RNA-induced silencing complex to the complementary region of the target mRNA. And this RNA-induced silencing complex then cuts the mRNA, leading to its degradation and silencing of the target gene. The small nucleic acid market is growing fast, and not just in rare disease anymore. And global oligonucleotide drug sales are projected to reach about $60 million by 2035, up from just $3 billion in 2020. Metabolism and the CNS diseases are the two main therapeutic areas right now. And you can see the key targets here. For example, PCSK9, AGT, APOC3, and others. Humanized mice carrying the full human gene sequence and allow the target to be expressed under its natural regulation control and ensuring the physiological levels and the tissue-specific patterns that faithfully reflect human biology. APOC3 is a novel lipid-lowering target. It raises plasma triglyceride levels by inhibiting LPL activity and blocking the clearance of TRL and their remnants. And targeting APOC3 reduces triglycerides and improves overall lipid profiles. The first approved drug reached market in May 2019 with several additional conditions currently in development. To evaluate small nucleic acid drugs targeting APOC3, we generated the humanized APOC3 mouse, in which the entire mouse coding region and the 3' UTR are replaced with the human sequence. Here, the PCR and ELISA confirmed stable expression of human mRNA and protein of human APOC3. And here we use this humanized mouse model to evaluate in vivo efficacy of a siRNA drug of Arrowhead on plozasiran. And the results show that a single dose of arrow APOC3 in humanized APOC3 mice significantly reduces the human APOC3 protein level and triglycerides over 31 days, with corresponding effects on LDL-C and total cholesterol. And this response is durable and statistically significant, confirming this model is suitable for RNAi efficacy studies and early drug screening. The part 4 is the applications in PK studies. PK studies are a critical part of preclinical development, as they determine the dose and dosing schedule for the clinic. In addition to efficacy and safety evaluation, humanized mice also play an important role in certain PK studies, particularly those involving antibody drugs. And FcRn binds IgG and albumin and protecting them from lysosomal degradation and releasing them back into circulation at neutral pH. And this recycling mechanism gives IgG drugs their long serum half-life. So antibody drug often has a long half-life compared to small molecules. Or during antibody engineering, the Fc region is frequently modified, such as through YTE mutations to strengthen FcRn binding and further extend the half-life of the antibody drug. However, mouse FcRn exhibits different binding affinity for human IgG compared with human FcRn. Wild-type mice cannot reliably predict human antibody from PK, and the Fc-engineered variants often show no effects in wild-type mice, while non-human primates are costly and challenging. So that's why we need FCR in humanized mice. In this model, it was generated by inserting chimeric cDNA sequence targeting human FcRn into the mouse Fc-GRT gene locus. And Western blot confirmed human FcRn expression in the humanized mice, but not in wild-type mice. And in PK studies with the IBI-112, an antibody, an anti-IL-23 monoclonal antibody, the humanized mice showed a serum half-life of 230 hours for the human IgG1 backbone, which extended to 340 hours with the YTE mutation. And this Fc engineering effect was observed specifically in the humanized FcRn background and was absent in wild-type mice. Therefore, the humanized FcRn models enable more reliable prediction of human PK for antibody drugs, and including Fc-engineered variants. To summarize, beyond the cases discussed above, for ready-to-use therapeutic target humanized strains, we have developed over 1,000 models, including single gene humanized, double gene humanized, triple or even 4 or 5 gene humanized. And we can divide these large collections of humanized models based on the type of therapeutic targets or the type of therapeutic modalities. For instance, we have more than 250 kinds of immune checkpoint humanized models, more than 170 double humanized models tailored to meet the preclinical research needs for bispecific antibodies. and more than 190 cytokine receptor humanized models that are widely used for autoimmune disease therapy development, and many others. And moreover, we also provide a full range of service, including model development and validation, drug screening, and efficacy evaluation. Thank you for your attention, and questions and discussions are welcome.
Great. Thank you so much for the wonderful presentation. We've got some questions in our Q&A, and if anyone else in the audience has more questions to ask, please put them in the Q&A and we'll address them now. Uh, so to start off here, we have a question about humanized mice being a substitute for NHP studies. So the question here is, are the knocked-in genes expressed in all target tissues that the normal target would be expressed in an NHP? Or is it limited to a subset of tissues? And if it's limited, does that change the toxicity readouts? So in other words, is the gene expression in the humanized mouse global or just restricted to particular tissues?
Yeah, now I can answer the question. And actually, different models have different advantage. They all kind of models have its own limitation. For the humanized mouse model that we established, normally we just replace the coded region, keeps the 3' 5' UTR. So the humanized gene have the similar expression profiles as the mouse endogenous gene. And but sometimes the human protein and the mouse protein have the same expression profile, sometimes may have different. But before we put on our human and mice on the market, normally we will do a completed validation, make sure that the human protein can reflect the expression profile as human body as best. So that's kind of a limitation. But the sum of gene, for example, the CD3 EDG human and mouse. So CD3 only expresses on T cells. So that's why you can use this mouse for all the toxicity and the PK studies for all the T cell engager molecules. Actually, one of our clients in China, they got the R&D approval right away with the mouse study. They didn't ask even NHP toxicity study, GLP studies. So which means that the mouse data can reflect the fully toxicity data.
Great. Thank you
Yeah.
very much. So another question we got here is, could you address the various humanization strategies we use, be that cDNA knock-in, exon and intron replacement, partial versus full knock-in, and how do you choose that on a case-by-case basis?
Okay, sure. Yeah, so actually, for different humanized mouse models, we use probably different strategies. It's always different because the regulation, the expression file sometimes are same in human and mouse, sometimes are different in mouse and human. Normally, we will design multiple strategies to humanize the special target, either cDNA, sometimes the genomic DNA, sometimes the 100% fully human sequences, sometimes just the extracellular domain. We normally investigate the expression and regulation of the target gene. Then we choose the best strategy to humanize this gene. And after we make several different kind of strains targeting the same gene, and then we validate all the strains before we start to use that for drug efficacy study. So it's if you have any, because we have over 1,300 different strains, really hard to tell how we decide the specific strategy for each gene. If you are interested in some gene, we can share more information with the client.
Great, thank you. And kind of leading from that, so for genes where we replace the 3' UTR, like in the humanized ApoC3, are there trade-offs when you swap out the mouse regulatory elements for the human?
Well, I'll answer the question. It's a good question. The 3' UTR contains regulatory elements that control mRNA stability and translation. For ASOs or siRNA, targeting engagement often depends on these non-coding sequences. So without the full 3' UTR, you may miss critical binding site or misrepresent knockdown efficacy.
Great. That's good to know. Yeah, so it really is case by case depending on which modality you want to use and what you need to be expressed. Okay, great. Here's another question. So, what experience does SMOC have with testing multi-specific antibody formats in humanized mice? We talked about T-cell engagers. Do we have other experience?
Yes, we test, for example, the PD-1 VEGF double humanized the mice used for the PD-1 VEGF bispecific antibodies, which one of our clients already got a clinical approval for their molecules. And also we try some of the triple specific humanized mice, for example, CD2, CD3, and also some EpCAM, triple specific antibodies. We have a lot of experience in testing those bispecific or multiple specific antibody molecules before.
Okay, great. Next, I'm going to summarize a couple questions kind of into one, which, because we're using in Inoculum, we're using the mouse regulatory elements most of the time, and we're including, human peptide sequence. Are there any expression difficulties that we encounter when generating these models?
Yeah, sometimes. So that's why you cannot humanize all the mouse genes with a mouse UTR. But we will try our best. Maybe if we adjust, maybe we can use human UTR, maybe 5' UTR, human 3' UTR like a mouse, maybe combination. Whatever, we try our best to adjust the expression profile of the humanized gene, just as good as a human gene in the human body.
Okay. And following up from that, for proteins that require post-translational modification for either regulation or function, do we see any challenges to that in the humanized mice?
Actually, it's really a good question. It's kind of hard to tell because the glycosylation, the post transcription modification, kind of different in human and mouse. If your molecule is definitely targeting the post transcription, post translation modification, maybe you have to maybe humanize all the whole system for the modification. For example, some glycosylation process. It's really hard to tell. But sometimes if the post-translation modification changed for some protein, for example, the glycosylation is lost during the in the oncology in the tumor, you can target the wild type, the just amino acid backbone. So it might be easier for us to study. It's always case by case.
Okay, great. Now we have some questions focused towards the CD3 EDG model. So first off, why did we select the black 6 background for that model?
Uh,
Um,
Actually, oh, okay, Dr. Lee, go ahead.
Uh, okay, normally the B6 is the most widely used inbreed strains for immuno-oncology studies. It has well characterized immune responses and is the standard background for syngeneic tumor models like MC38 and B7 and B16. And we also offer the CD3 humanized models on the BARBC background, which supports additional syngeneic models such as CT26. And having both backgrounds allow you to evaluate T-cell engagers across different tumor microenvironments and validate findings in more than one setting before moving to the clinic.
Okay, great. And still working on that model. Someone wants to know, there's several different strains on the market of humanized CD3 EDG. Does our model have any advantages?
Uh, uh, I think it's very hard to tell really advantage or not. because the strategy and everything verified as long as the expression pattern is quite stable. We uh,
Yeah,
we're not easy to claim very obvious, but from the case study, we have a lot of historical data to prove that it works very well. Probably Dongxiao can have further
yeah, I
comment.
can address that questions. Yeah, because from our strategy, you can see that we humanize all the CD3 EDG genes in one backbone. So that's why all the CD3, the three subunits, CD3E, CD3G, and CD3D, have the same expression profile and the same expression level in one mouse. For other companies, probably you can check their website. You can see the expression, their validation data on their website. They can only show the CD3E expression in their mouse model. No company have shown the CD3-EDG expression profiles in their website. Because I think the strategy that they make those mouse models, they make three different transgenic mice and cross them together, which means that the three subunits cannot be expressed at the same level. They may be regulated by the same promoter, but if they are integrated in different chromosome location, the expression level of the three subunits might be different. So that's why they cannot show the expression of the three subunits in their website. I think which makes the difference for our CD3 EDG human mice compared with other companies.
Great. So next up, we have some more technical questions. So if we have two knock-in lines that someone's interested in potentially crossing together, is that something we can support, and what's the time frame for doing that?
Normally, so far we have put 7 human genes in one mouse. Of course, we can do customized humanization for the client. If we already have the single humanized mouse model, we can always cross them together, but to make sure they are on different chromosomes. If not, if they are on the same chromosome, we have to do another humanization using one of our single humanized mouse model. Probably for this kind of study, it takes at least 9 to 12 months to get the double homozygous humanized mice. If they are on different chromosomes, and make sure it takes 6 months to get a double humanized mouse with homology cohort for efficacy study. For triple, probably you have to double the time, probably 2 years to get the triple humanized mice. This kind of an estimated timeline, but always case by case. If you have any genes you are interested, contact our BD managers.
Okay, great. And then on the other side of that, if there's a humanized gene that we don't have, what's the process and timeline to generating that new model?
So normally, if we can use a simple way, for example, the cDNA humanization with CRISPR-Cas9 technology, we can guarantee to deliver the heterozygous humanized mice to the client in 9 months. But normally, the mice can be delivered in 6 months. But if kind of a very complex gene, we cannot use CRISPR-Cas9 technology, then we have to go back to ESL homologous recombination method. The timeline will be 12 to 14 months for the F1 heterozygous mouse model. And for homo mice, 3 months will be extra time.
Okay. Next, we have some questions about particular use cases. So obviously, Dr. Li talked about using the humanized FcRn mice for doing PK studies. Are there other strains that we'd recommend for PK work?
Yeah, that's a good question. So rats are widely used in PK studies because they allow larger blood volume than mice, and which makes the serial PK profiling, for example, multiple time points from the same animal. And so we also have a range of humanized rat models, including the FCRN humanized rats, which can be used and has already been validated.
Okay,
Yeah,
great.
also the albumin is also another factor that can affect the PK data. We have the human albumin, serum albumin humanized rat, and also the double humanized rat for FcRn and albumin. So you can mimic the PK study in a rat more precisely with human, like human.
Yeah, to add a bit, we have a variety of tools. So it really depends on which PK question we want to answer. For example, the humanized FcRn mice is good to study the antibody recycling and the Fc engineering. However, if we want to understand the baseline clearance, we also have a FcRn knockout mice. If we want to understand how well the drug behave in the human IgG environment, we also have a human IgG transgenic mice to understand the drug competition PK. Sometimes we want to understand the effector function PK. We have the Fc gamma receptor humanized mice. It really depends on the what kind of PK question we would like to answer.
Okay, great. Thank you. Last the question here we have is why not just use humanized the immune system humanized mice instead of using gene target humanized mice for doing T-cell engager and checkpoint inhibitor work?
I'll answer the question. It's really a good question, and the human immune system mice can work for T-cell engagers, but they come with donor variation, and graft versus host disease risk and higher costs, as I mentioned above. And more importantly, for some immunocheckpoints like CD47, the macrophages are missing and poorly reconstitute in a human immune system mice. Instead, the gene-humanized target models can solve this by putting the human target into immunocompetent mouse.
Okay, makes sense. Actually, we just got a couple more questions in. These are related to the nucleic acid drugs. So first off, is there any difficulty with targeting, the humanized version of the gene with a nucleic acid drug relative to the mouse version? So is there any expression difference that would alter how the test articles work?
I think we cannot simply tell work better or not because if you use the mouse gene, you need we still call surrogate uh, drug. You need to use a mouse sequence, but use humanized mice, you can directly use the human sequence that you develop the human drug directly after you get this efficacy or other PK or other pharmacology parameters. It's more translational to human clinical readout, and also you don't need to redesign the test article. I think that's a major advantage. Um,
Okay. Another question on nucleic acid drugs. How does SMOC address the challenges of delivering oligonucleotide drugs to the central nervous system?
I think the main difficult to deliver to the central system is the blood-brain barrier. And I think for this you have several strategies. You can use the TFR1 humanized mouse by link the drug to the TFR1 antibody and to across the brain blood barrier. And another way is to use a different administration, like IT administration. Yeah.
Great, thank you. I think that's all the questions we have from our audience so far. Do we have any final closing words from our panelists?
I think here I want to emphasize because a lot of people want to know uh, the humanization strategy, cDNA or the full sequence or use the human regulatory or use the mouse regulatory. So here, first, we want to know exactly the function of the gene and also your treatment. And uh, for example, if you use like the regulatory small nucleotide drugs, probably we will include the intron, include the upstream and downstream regulatory sequence. If it's type 1 transmembrane protein, we usually just use the chimerical protein, only humanized extracellular cDNA. And also the beauty of this company, we can knock in like a 150 kb or even larger fragment. Based on that capacity, we can sometimes, if that's new needs, we can design those strategy for you. And also, interesting topic is some gene, I think more than 90% human and mouse has a similar expression pattern and a similar function. Those for those genes, we prefer use mouse regulatory elements so that keep the expression pattern, the concentration, everything similar as mouse. Because anyway, we do this study in mouse physiological condition. But some gene in human and mouse, they have different expression pattern. Our mouse mm, doesn't have that counterpart of gene. So in that case, we rather just knock in a very large fragment, include the intron, include the regulatory element into the mouse safe harbor, so that can regenerate the human expression pattern. So that's one kind of powerful this very large fragment knock-in.
Great. Any other comments from the panel?
Yeah,
Yeah.
I also that's another very interesting question about the post-translational modification or those stuff. I also read some literature. Between mouse and human, we cannot very clearly say what's the difference. There's no clear theory, but I think that's more like a dynamic. Nowadays, there's a lot of publication, even in our human body, the same gene at a different environment or different physiological pathology condition, they have different post-translational modification. So that part, I think, is not because we put the gene in mouse, the post-translational modification is a concern. It's generally speaking, is very dynamic, even in human patient. Uh, that's my comments about that part. Lei?
I was asked several times. Basically, people ask you have a different target gene humanized mice. You also have a CD34 positive human PBMC reconstituted mice and also human immune system mice. So which one should I choose? So my answer is it depends on what question you want to answer. If you are answering a early discovery question, for example, you are testing your format, your antibody affinity, or your FC design, I would use the target humanized mice. However, if you want to answer the question, how well does my drug behave in the human immune system? So the human immune system mice is better. That's a very straightforward answer.
Yeah. So as Dr. Jing and Dr. Chen said, that every drug has a different situation. If you are really interested in using the humanized mouse model to test your drug in vivo, and as you know that we have very knowledgeable team, and all of us have many years of experience working with in the industrial, in the preclinical studies, and we will form a service team for your project, and we can always discuss to try our best to meet your final requirement.
Great. Well, thank you so much to our speaker, Dr. Li, to our panel, and also to our audience. And thank you for the fantastic questions. We're going to close this webinar for today. But before we leave, I want to let you know that we'll be having a webinar next month entitled Humanized Mouse Models in Autoimmune Disease Research. So please be on the lookout for invitations to that. We look forward to hosting you at the next one. And everyone have a great rest of your week, and we'll see you at our next webinar. Have a great day. Thank you so much.